Hi, hello, welcome to UCrack Academy. Today we are going to see about Principles of Inheritance and Variation Part 2. So the first topic is about chromosomal theory of inheritance. Have we know that the chromosomal we have 23 pairs, but here we are going to see who have proposed here. Walter Sutton and Theodore Boveri in 1902. So they worked out the chromosome movement during only the meiosis time. And even though the behavior of the chromosome, it will maybe have changes with the gene or otherwise with the movement of the chromosomes, we can explain with the Mendel's law. So Sutton united the knowledge of chromosomal segregations with Mendelian principles like chromosomal theory of inheritance. Chromosome and gene are present in pairs in diploid cells. Even homologous, like these are the gamete formations happens only with the meiosis. And fertilizations also is happen, restores the chromosome number to diploid condition. The chromosomal theory of inheritance claims that is the only segregation asserts independently. What is the experimental variations of chromosomal theory? Here have introduced this Thomas Hutton Morgan and his colleague. Morgan worked with the tiny fruit flies like Drosophila and Melonganster. Why Drosophila? Here suitable genetic studies. Even though it is a simple synthetic medium in the laboratory, even complete life cycle is only 2 weeks, and a single mating produce a large number of progeny fly. Clear differentiation of male and female flies. Have many type of hereditary variations that can be seen with low power microbes or microscopes. You can see through that. Linkage and recombination. How we can recombination the genetic materials and how we can interlink and what can type of a thing we can get it, that we will see. Here the few scientists have introduced Morgan hybrids yellow bodied, white hide females to brown body, red hide male and intercrossed their F1 progeny. He observed that two genes did not segregated independently of each other and the F2 ratio deviated very significantly from 9:3:3:1 ratio. When two genes in a dihybrid crosses were situated on the same chromosomes, even though parent gene combination higher than the non-parental type. Morgan distributed due to the physical association or linkage of two genes and coined the term linkage. Linkage, here physical association of gene. Recombination means non-parental gene combination. Morgan found that even when genes were grouped on the same chromosomes, some same gene very tightly linked, that is called very low recombinations. If it is a loosely linked in the sense, showed higher recombination. The gene white and yellow were very tightly linked and showed 1.3% recombination. The gene white and miniature wing showed 37.2% recombination, hence loosely linked. Alfred and used to the frequency recombinations with the gene paired like the measurement distance gene mapped their position on the chromosomes. Polygenic inheritance. Humans have no distinct tall or short, instead a whole range of possible heights. It may vary depends on the parent. Such traits are generally controlled by three or more genes are called polygenic trait. Besides the involvement of multiple gene, polygenic inheritance also takes into account the influence of environment. Even human skin color also is another classical example of polygenic inheritance. In her polygenic traits, the phenotype reflects the contributions of each allele, like that is the effect of the each allele is additive. You can assume the three gene, like you can take any alphabets, but here I have given you A, B, C, control the skin color in human. But the gene type is not the same, gene will not give, that is different actually. Pleiotrophy. A single can be exhibit the multiple phenotype expressions. Such gene is called a pleiotropic gene. The mechanism of pleiotropy in the most cases is the effective of the gene metabolic pathway which contributes towards the different phenotypes. So this is the human disease which is called phenylketonuria. This disease is actually a mutation. If it is a code, ஒரு செயினில் வந்து கோட் மாதிரி கண்டினியூஸாக போயிட்டே இருக்கும் போது அதில் இருக்கிற ஒரு ஜீன் மட்டும் if it is a repeated or it is the different way compare with the parent, no, that is actually a mutation. So that we can say it is a enzyme phenylalanine hydroxylase. Phenotypic expression characterized by mental retardation, otherwise you will get reduction in hair, otherwise reduction in skin pigmentation. Sex determination. Henking 1891 traced a specific nuclear structure during the spermatogenesis of some insects. 50% of the sperm received from the specific structure. 50% of the sperm did not receive it anyway. Henkin gave a name to this structure as the X body. X body of Henkin was later named as the X chromosomes. Sex determination of grasshopper. This is XX minus XXO type. But here also all X bears only X chromosomes, which is called autosomes. 50% of the chromosomes X, 50% do not. And even the egg fertilized with the sperm with X become female 22 plus XX. Egg fertilized with the sperm without X chromosome become male 22 plus XO. The next one is the same mammals, insects types, how it can be determined XX, XY type. Both male and female has the same number of chromosome. Even autosomes is there. That is A plus A and XX. Male you have a large X chromosomes and one very Y chromosomes, capital A plus XY. This is called male heterogamy and female homogamy. Sex determinations in birds. Female birds have two different sex chromosomes, Z and W. Male bird have a similar sex chromosomes called ZZ. Sex determination, female as a heterogamy and male homogamy. Here it's about the honeybee. You can see here also the individual receivers. Everything was similar. The number of the pattern is differs. Here also the offsprings from the fertilization of the sperm and egg also developed from the queen and worker bee that you have known from the female. Unfertilized egg develops as a male drone, which means parthenogenesis. The male have half of the number chromosomes than that of female. Even diploid, 32 chromosomes, and males are haploids, 60 number of chromosomes. So haplodiploid sex determination system. Here also the same, that male produce sperm by the mitosis and do not have a father and thus cannot have sons, but they have grandsons. Mutation, which is most important topic. Mutation is the phenomenon which results in alterations of the DNA sequence. So the genotype and phenotype is there in the two. So that we are going to see in the detail in the next chapter. In addition, recombination, mutation also know is another phenomenon of the variations which can be seen by DNA. Loss, deletion or gain, insert or whatever it is happening through the chromosomes where that it's moving through on. Since the genes are located only on the chromosomes, alteration in chromosomes results in abnormal. Chromosomal aberrations are commonly observed in cancerous cell. Mutation also arise due to change in a single base pair of DNA. That is called point mutation. Example, sickle cell anemia. Deletion and insertion of the base pair of the DNA causes frame shift mutation. Genetic disorders, pedigree analysis. So which is a several generation of a family which we have now. So here the particular traits is representing in the family tree over the generations. If you look at the male, female, sex unspecified. So affected individual, mating relatives, and then if you look at here all together here, the children's and below birth to the right. So if you look at the male child affected with the disease, so 5 unaffected offsprings will be occur. That is pedigree analysis. Autosomal dominant. Look at the pictures here. The same pedigree. It is also affected at least one affected parent. And it is also every generation it will be happen. 2 unaffected parent only have unaffected offsprings. Traits are controlled by dominant gene. Both male and female are equally affected. Traits do not skip generation. That is called the tongue rolling ability, etc., which we can say. This is also continues with the recessive pedigree, the autosomal, which is also affected by the offspring. Traits also the dominant. It is also homozygous. Both the male and female equally affected. Traits may skip generations. 3:1 ratio between normal and 2 affected. Appearance of affected children from normal parents. All children or affected parent are also affected. That is called sickle cell anemia. Mendelian disorder. Genetic disorder grouped into 2 categories. Mendelian disorder, chromosomal disorder. This is actually the disorder determination by the alterations or mutation in the single gene obey the principal inheritance during the transmission from one generation to the another. It also can be expressed in the pedigree analysis, hemophilia, color blindness, cystic fibrosis, sickle cell anemia, phenylketonuria, thalassemia, hemophilia. In this disease, single protein like a part of the cassidy and the proteins clotting of the blood is affected. So what will happen? The blood flow will be stopped. Sex-linked recessive disease, unaffected carrier, female to some other to male progeny. The same it is opposite. From the mother will get it at least. Affected transmitted the disease only to the son, not to the daughter. Daughter can receive the disease from both mother and father. Sickle cell anemia. Like no, glutamic acid by valine at the sixth position of the beta globin chain of the hemoglobin molecule. Substitution of amino acid takes place like GAG and GUG. This may be variant. This is also comes from the RBC, and also it is can be the sickle-like structure, autosome linked recessive traits, and heterozygous. This disease is controlled by the single pair of allele. There are the three I have given you: HBA, 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 HBS, and HBS, HBS. But this is both are phenotypes. This two comes under that is heterotypes. Phenylketonuria, autosomal recessive traits, inborn errors of metabolism. It is also affected by the phenylalanine hydroxylase that converts the amino acids phenylalanine to the trypsin. This you have to memorize, no other option. In the absence of the enzymes, everything will be accumulated and it will also converted into the acid and other derivatives. Accumulation of these results in mental retardation. This derivation excretions through kidney. Chromosomal disorders, which is very dangerous actually, due to the absence or excess or abnormal arrangement of one or two chromosomes. Failure, autophyloid, polyploid, that is cytokines failure, trisomy, which is 2n plus 1, that is the additional copy of a chromosome may be included in an individual's. Monosomy, and individuals may lack one of any one pair of chromosomes. Down syndromes. This is actually caused only the trisomy of 21 pairs. And this is Langton Down 1866. They have found it out. These are the symptoms you can see in these pictures. Short statures with a small round head, proud tongue, partially opened mouth, Palm is broad with characteristics palm increased. Physical, psychomotor, and mental development is retarded. This is also the same 47 chromosomes which is appears here, but that is karyotype. Turner syndromes. This is also caused 44 plus XO. Overall, the masculine development and then development female characteristics and individual steroids. Here's also the ovaries and a lack of other secondary sexual characteristics. Everything may have difference. Followed by few questions. Question number 1. In Mendel's experiments on pea plants, that traits that disappears in the F1 generation but reappeared in F2 generation is called A. Dominant traits, B. Recessive traits, C. Codominant traits, D. Linked traits. Answer B. Recessive traits. Question number 2. The term homozygous refers to an individual that is option A, two different alleles for a particular gene. B, two identical alleles for a particular gene. C, multiple alleles for a particular gene. D, no alleles for a particular gene. Answer B, two identical alleles for a particular gene. Question number 3. The genetic makeup of an individual is referred to as its A. Phenotype, B. Genotype, C. Allele, D. Homozygosity. Answer B. Genotype. Question number 4. The observed physical characteristics of an individual are referred to as A. Genotype, B. Phenotype, C. Allele, D. Dominance. Answer B. Phenotype. Question number 5. Which of the following is an example of a sex-linked inheritance? A. Blood type, B. Eye color, C. Height, D. Color blindness. Answer D. Color blindness. Question number 6. The inheritance of traits controlled by a single gene with two or more alleles called A. Incomplete dominance, C. Codominance, C. Multiple alleles, D. Polygenic inheritance. Answer C. Multiple alleles. Question number 7. A gene located on the X chromosome is called an A. Autosomal gene, B. Recessive gene, C. Dominant gene, D. Sex-linked gene. Answer D. Sex-linked gene. Question number 8. In a dihybrid cross, if two genes assort independently, the phenotype ratio in the F2 generation will be A. 3:1, B. 1:2:1, C. 3:3:1, D. 1:1. Answer C. 9:3:3:1. Question number 9. The phenomenon where a single gene influences multiple traits is called A. Incomplete dominance, B. Codominance, C. Philiotropy, D. Polygenic inheritance. Answer Philiotropy. Question number 10. In human, the inheritance of A, B or blood groups follows the pattern of A. Complete dominance, B. Incomplete dominance, C. Codominance, D. Multiple alleles. Answer C. Codominance. Question number 11. The inheritance of skin color in human is an example of A. Complete dominance, B. Incomplete dominance, C. Codominance, D. Polygenic inheritance. Answer D. Polygenic inheritance. Question number 12. The presence of an additional copy of a chromosome in a cell is called A. Polyploidy, B. Aneuploidy, C. Deletion, D. Duplication. Answer B. Aneuploidy. Question number 13. Down syndrome caused by an extra copy of chromosome 21 is an example of A. Polyploidy, B. Aneuploidy, C. Deletion, D. Duplication. Answer B. Aneuploidy. Question number 14. The process of exchange of genetic material between homologous chromosomes during meiosis is called A. Crossing over, B. Inversion, C. Translocation, D. Non-disjunction. Answer A. Crossing over. Question number 15. 8 traits that overpower and hide another traits is called A. Overpowering traits, B. Complex traits, C. Recessive traits, D. Dominant trait. Answer D. Dominant trait. I hope you all have got an idea about this, children. If you want to know more videos, kindly subscribe the channel and hit the bell button. Thank you so much.